that I did with E-block, I did not like. For lots of reasons. So many tabs. Okay. So, in the process of breaking down glucose, whether or not you have oxygen, whether or not you have mitochondria, you are going to do the same thing. You are going to do glycolysis, whether you are a prokaryote or you are a eukaryote that does have mitochondria. Regardless of if oxygen is present or not, the first step in the breakdown of glucose is always glycolysis. Glycolysis happens in the cytoplasm, so you don't need organelles in order to do this. You don't need a membrane-bound organelle. So anybody can do this, any kind of cell can do this. So no matter what glucose shows up, the very first step in the breakdown of it is going to be glycolysis. Glycolysis, as a reminder, breaks glucose in half. Glyco meaning sugar or sweet, lysis meaning to break or split. So we break glucose in half. It is oxidized, which means what? It loses electrons. Sydney, what's up?
Wait, is this only autotrophs that can do this?
Nope, this is all cells that need to get ATP from glucose. So autotrophs that make their own glucose could do this with their glucose that they made. Heterotrophs can do this with the glucose that they consume. Anything that is needing to break down glucose to make ATP. So the glucose is oxidized. If it loses electrons, who picks up the electrons? NAD plus, which becomes NADH once it picks them up. So NAD plus is reduced, it gains electrons, and becomes NADH. We also, when we break this molecule in half, it drops its electrons, that get picked up, and we make these two pyruvate molecules. We make a little bit of ATP, but not a whole lot. In order to do that, we need ADP and some phosphate. Where would we get ADP and phosphate from in the cell?
Mitochondria.
Mitochondria usually is making ATP. What happens to ATP? It loses phosphate when it goes and powers proteins. And so once it has given the energy that it contained to a protein to do something, we get back ADP and phosphate again. And so we can reuse that ADP and phosphate, put it back together to make a new ATP. All right, so we need ADP and some phosphate. We need NAD+. What we don't care about at this point is oxygen. We do not care if it is present or absent. Okay, so the reactants that we need in order for glycolysis to occur, we need glucose. We need an initial energy investment, which wasn't represented on the previous slide, because often we just kind of say, well, it makes 2 ATP. Realistically, we spend 2 ATP to make 4 ATP, so it is a net gain of 2 ATP. We need some NAD+ to pick up the electrons that glucose loses, and we need some ADP and phosphate in order to make ATP. All right, so if a glucose molecule shows up, we have this. We need these things in order to perform glycolysis. With me so far? Okay. After that point, what happens next depends on if oxygen is present or not. And really this should have an asterisk that says, and if you have mitochondria. Because if oxygen is present but you have no mitochondria, you can't do the citric acid cycle. You can't do pyruvate oxidation because those things happen in mitochondria. So if you have mitochondria and glucose and oxygen, then you can do all of the stuff that we talked about yesterday. Glycolysis is always the first step. If you have oxygen and mitochondria, you can go forward and do pyruvate oxidation and the citric acid cycle and the electron transport chain. You can go through all of those steps. But if you do not have oxygen, you can't do all of that. That's not going to work. After glycolysis, your option is fermentation. That's what you're going to do. Even if you have mitochondria, if there's no oxygen, you cannot do all of those things. Your only option is fermentation. If you have no mitochondria, but there is oxygen present, can you do these things? No, because you need mitochondria for those two. So no mitochondria, you're always going to go to fermentation. Mitochondria, no oxygen, you're going to do fermentation. No oxygen, no, hang on. Yes, oxygen and mitochondria, now you can unlock all of that other ATP that was locked up in that glucose molecule. Okay, so always start with glycolysis. From there, it's a choose your own adventure to a certain extent based on if oxygen is present and how many or what type of organelles you have. Okay, So, which process is this? Is this going to be my aerobic steps or my anaerobic? Aerobic. These ones don't use oxygen yet, but these things only happen if oxygen is present. So these are my steps to aerobic respiration before the very final step. We make a bunch of NADH and FADH2. We make a bunch of loaded up electron carriers. What happens to them in this process?
Wait, what happens to the electron carrier?
Yeah, where do NADH and FADH2 that we just loaded up with electrons and hydrogens, where do they go? The electron transport chain and ATP synthase, right? They take all of that energetic stuff. They take the electrons and the hydrogens to the electron transport chain and they drop it off with those proteins. Those proteins use the electrons and they pump hydrogen ions, and then those hydrogen ions power the ATP synthase. Where do the electrons end up at the very end of this process? They go through all the proteins and then they're all used up. Who takes the electrons at the end?
The hydrogen?
Oxygen. The oxygen. Remember, in our cellular respiration equation, we have glucose, or aerobic cellular respiration equation, I should say, and oxygen makes 6 CO2 plus 6 H2O, and there's ATP involved in this, but these are the inputs and outputs if we're simplifying. This gets broken down in all of these processes. My glucose gets broken down and makes 1, 2, 3, 4, 5, 6 CO2 molecules. What this does not show you is the last step where oxygen comes and takes the electrons and some hydrogens and becomes water. So oxygen is my final electron acceptor in this process. Oxygen is the thing that stops me from having a traffic jam at the electron transport chain. I'm going to go back to some of the, not that one. No, I at you.
Is there like a video that like you can actually, that zooms in enough that you can actually like see the electrons going in and out?
There are models, but not like an actual video. So you can, there's animations, but not really like a real video. So this video that we watched yesterday, so this is our electron transport chain, remember? Those are our proteins that are using the power from the electrons to pump hydrogen ions across the membrane that will then flow back through ATP synthase. This is the thing that's going to make ATP, but now my electrons are at the end of the transport chain. Somebody has to come pick them up so that the next time another electron carrier comes and drops off electrons, I don't have a traffic jam. Somebody needs to take these away so I can keep putting new electrons through this process. Oxygen is our buddy who does that. Oxygen comes, takes our electrons, takes a couple of hydrogen ions, and becomes water. This is where all of the electrons end up in aerobic respiration. So we have a place for them to go at the end of that process. Okay, so all of those electron carriers that we made, I'm going to small screen this. All of the electron carriers that we made in glycolysis, in pyruvate oxidation, in the citric acid cycle, all of those go and get used. They drop off their electrons at the electron transport chain, and oxygen picks them up at the end of it. At the end of the ETC, it becomes water. That's where our water waste is coming from in aerobic respiration. After our electron carriers have dropped off their electrons at the transport chain, after NADH drops off its electrons, what does it become? NAD plus. It goes back to being NAD plus. It's an empty shuttle now. Okay, so all of these things that we built up, we get back our FAD and our NAD plus after they've dropped off their electrons they were carrying, so they can go back and participate in the citric acid cycle, or pyruvate oxidation, or glycolysis. We have those electron shuttles that have now emptied themselves that could pick up something new, and we can go through this process with another glucose molecule. This is possible this transport of electrons, dropping them off, and then going back to the process is only continually possible because somebody is taking those electrons away. If they didn't, we'd get clogged up and we just have this buildup of these loaded up shuttles with nowhere to drop things off. It's like if you had a whole line of buses that were trying to drop people off at the one stop and you just run out of space. If you keep loading buses, that's not going to work. You're not going to keep moving people through this system, right? You have a traffic jam. With me so far? Questions? Yes.
This is in, no, never mind.
This is aerobic respiration.
Yeah.
So this is happening, glycolysis is in the cytoplasm. The rest of the steps are happening in mitochondria. But this is cellular respiration, specifically aerobic respiration because oxygen is getting used. All right, and this happens in anything that has mitochondria and oxygen, which could be a plant using its own glucose, it could be an animal, it could be a fungus, it could be a protist. As long as you've got mitochondria and oxygen, you could do that process. All right, so in a situation where there is no oxygen, if we are in an anaerobic situation, we have a problem because there's nobody to pick up the electrons at the end of this process. All of these things create a bunch of the full shuttles. If there's no oxygen, there's nobody to pick up the electrons at the end of the transport chain, we get a traffic jam, and we stop this process of making ATP. What that means is we don't have any NAD+ to do glycolysis either. We keep loading up our shuttles, and we get rid of the NAD+ because it's become NADH. If there is no NADH, then we can, or if there is no NAD+, because it's all NADH, we cannot do glycolysis again. So the whole point of fermentation, whether it is lactic acid fermentation or alcoholic fermentation, the whole point of that part of the process is to give you back your NAD+, to give the NADH somewhere to offload their electrons that they were carrying. So our first step in breaking down glucose, always glycolysis. In that process, it uses NAD+ as a reactant. It makes NADH as a product, among the other things that it makes. Fermentation gives us back the NAD+. It oxidizes NADH, allows it to lose its electrons. It also makes some waste products. But the important thing is that we get this guy back, so that the next time a new glucose molecule comes, we can do glycolysis and we can get our 2-ATP out of this. Depending on what kind of organism you are, depending on what kind of enzymes you have, you are going to do one of these two types of fermentation. Organisms don't do both. They either do lactic acid fermentation or they do alcoholic fermentation. And it's just purely based on your genetics. What enzymes do you have and how are they breaking down the pyruvic acid that you made? So things like plants and yeasts, and I was looking it up and the internet was being weird with the Cloudflare downage. I can't find a specific plant that does this. Most of the plants that we ferment in alcoholic fermentation, it's actually yeast on the plant that is doing the fermentation. Like if you're talking about grapes and wine or barley or corn or all of the things that people use to ferment to make alcoholic beverages, it's because there is yeast also. So yeast is really the big one that does alcoholic fermentation. There are a couple of very specific locations in some plants that they can do fermentation also. But in alcoholic fermentation, it makes two waste products. It makes ethanol and it makes carbon dioxide. What it does not make is ATP. Fermentation alone does not make ATP. Fermentation and glycolysis make 2-ATP because glycolysis makes 2-ATP, but this process alone does not make ATP. The only point of it is to give you back your electron carriers. These things are trash. These are waste. We don't actually care about these. Same thing in lactic acid fermentation. We actually don't care about having lactic acid. That's not really the goal. That is a waste product that is produced at the same time that we are oxidizing NADH to get NAD+, but we also get some trash at the same time. Yes?
So what are the products of fermentation?
Depends on which type.
NAD+ or H?
It is the NAD+, and then if it's alcoholic fermentation, these things. And if it is lactic acid fermentation, it'll be this. The next slide has a picture of the process a little bit better. Yes.
Is this the same lactic acid that gets in your legs and your body?
Yes.
So what does it do to your body?
If it were to build up, it might be a problem, but your cells get rid of it really fast. It goes into your bloodstream and gets broken down. So ultimately, it's not doing anything bad to you. It is often credited for giving you muscle cramps. That's not really the case, unless you have circulation problems or like kidney problems. It is unlikely to be giving you issues. Your body's really good at breaking it down.
But what is it breaking down? Like
What is it breaking down?
what is being breaken down in that?
Lactic acid. Lactic acid is a 3-carbon molecule. It's just the name of that molecule. Whether you are something that does alcoholic fermentation or you're a different kind of organism that does lactic acid fermentation, the goal is regenerate NAD+. Get back NAD+. You just also happen to make some waste in the process. Oop, too far. Okay, so this is what it looks like for lactic acid fermentation. This is a simplified molecule. It does have some H's and O's and some other things attached to it, but it is a 3-carbon molecule. Lactic acid is also called lactate. And this is alcoholic fermentation. There is a little bit of breakdown of this molecule. We break a carbon-carbon bond in that process, and so we get 2 CO2 molecules, which we broke one of those off of each of those, and then we get 2 ethanol molecules. So we still have all of the carbon atoms that we started with. It's just that now we have broken them up and made them kind of different. This part alone, this is the fermentation part. Oftentimes, and I think they did this in the video, people lump together fermentation and glycolysis as one process. This is the fermentation part. This is glycolysis. Glycolysis can be partnered with fermentation, or it can be partnered with those steps of aerobic respiration, like we saw with the pyruvate oxidation and the citric acid cycle and the electron transport chain. If we're looking at just fermentation alone, not fermentation and glycolysis, how many ATP are being made?
0.
0. Just this part in this box, not glycolysis. 0. We do not make any ATP in the fermentation part of this process. The point of it is to give you back NAD+. So that the next time a glucose shows up, you can do glycolysis again and get your little 2 ATPs. Questions? Okay. So, summary. Point of fermentation is to allow NADH to drop off its electrons somewhere, to give it to somebody, because oxygen's not around or this organism doesn't have mitochondria to do aerobic respiration. The point of doing that is so that when a new glucose molecule shows up, you can do glycolysis again, because you have the NAD+, that is a reactant in glycolysis. So the reactant needed for glycolysis. All right. The cell can keep making ATP without oxygen as long as we can do this fermentation process, because we have something that is taking the electrons from NADH. We get NAD+ back. Now a new glucose shows up, we can do glycolysis again. This process, just like glycolysis, is happening in the cytoplasm. You do not need an extra organelle to do this. Sydney, did you have a question?
Yeah. Where does the ATP go after fermentation? Or
Wherever the cell needs it. It goes to power proteins and other cellular functions.
but what takes it there?
Typically diffusion. You could theoretically package things up into vesicles and move them around, but typically it's just they're bouncing around. You have this is happening all over the place in the cell. Cells have thousands of mitochondria at times. Like some cells have tons of mitochondria if they need a lot of ATP. They're sprinkled all throughout the cell. This glycolysis is happening in the cytoplasm. So this is happening all over the cell. So ATP is being produced and used all throughout the cell all the time.
But what if it doesn't have mitochondria?
Then this is happening all throughout the cytoplasm. Glycolysis is happening in the cytoplasm, which is that fluid-filled space around all of the organelles in the cell.
So the vesicles would take it wherever it needs to go?
Theoretically, yes, but more likely, you just have enough of the enzymes that are doing this process of glycolysis, or you have enough mitochondria sprinkled around that you don't really need to transport them, because essentially you've got ATP factories everywhere in your cell. So they're just like spitting out ATP that go out, and they just come in contact with something that needs ATP, gets used, converts it back to ADP and phosphate, and we can repeat that process with the ADP and phosphate. Put them back together through glycolysis and either fermentation or the rest of the aerobic process, and turn it back into ATP and do it again and again and again and again.
So if there's so many in the cytoplasm, how come it doesn't make as much ATP as
Aerobic.
as aerobic, but if it's more of it, then how come it doesn't have the same effect?
Ultimately, it's about efficiency. If I want to make a lot of ATP, I can take one glucose molecule and turn it into 30 some odd ATP, or I can take the same one glucose molecule and turn it into 2.
Got it.
If I want a lot of energy and I'm just doing anaerobic respiration, I'm going to need a lot of glucose. And so in aerobic respiration, you can take the same amount of glucose and make way more ATP than if you were just doing anaerobic.
Got it.
Other questions? Okay. That is the end of new stuff. It's the end of notes.